one of the things we learned during COVID, and we've learned since COVID, is that we need to work together and bring down silos that we have sometimes. And it's kind of the mantra of the talk I'm giving today. The other thing is that we meet, and generally speaking, I don't know about some of the younger folks in the room, I know from my generation, it's usually the coffee breaks, the poster sessions, and other times when you can actually talk to individuals that you start to really build the teams that need in the future. And so again, I encourage anybody to talk to some of the speakers afterwards and during the meeting to learn more. And you never know when those connections will come to your advantage some point down the line. All right, so just my disclosures, basically, I want to talk a little bit about the Galveston National Lab and not go into a lot of details, but it's a BSL-3, BSL-4 lab that we have as part of the network in the US. It's part of the regional labs, and then there's two national labs. One national lab is the Boston University, the National Emerging Infectious Disease Laboratory, and the sister lab is the lab in Texas, in Texas that we have. And so those are the two that have BSL-4 capability, and we have, at the Galveston National Lab, we have about 14,000 square feet of animal space and a laboratory space for BSL-4, plus we have an enhanced BSL-3, agricultural BSL-3, and BSL-3 labs for doing lab work. So it's a really nice infrastructure, and we do everything from preclinical or discovery to preclinical and non-clinical studies. So we actually, with a lot of these diseases that we're working with, at least in the US system, we have to have a non-clinical pathway to licensure, and that's the animal rule that the FDA has laid out for us. And so we carry out those studies in our laboratory to do that as well. So those are typically done with well-documented animal studies in animal models and regulatory requirements around that. But again, we do everything from basic to that kind of work. We also do a lot of training, so we have students. What's really nice about the BSL-4 labs that are done with the NIAID and NIH facilities is that they are actually in academic settings. So we have students, both postdocs, young students, and then we have graduate students. And we actually have folks coming to visit us. So it's an opportunity for anyone that wants to come work in these labs. Unlike the government labs in the US, it's kind of difficult to work in. These are actually a lot less paperwork for you to come work in our laboratories and actually do work with us. So again, an opportunity. All right, so my lab's just starting up back from industry. I left industry, came back into academia. And so I will say that this is a ChatGPT generated slide. I ask it what it should look like. But most of my work has been in viral hemorrhagic fevers, vaccines, therapeutics. I was a co-developer of ZMapp, the monoclonal antibody that was used in the West African outbreak. But I really focus on combinational synergies and looking at FDA-approved drugs, because when we have an outbreak of pathogen X, it's going to be very difficult to turn around and find a solution really quickly, even with our current concepts that we had. We were also working in 2010 on messenger RNA systems for these viruses as well. I also do some predictive analytics, mostly in my outside work that I do as a clinical trial statistical analysis team member. And then I'm looking at some synthetic biology, mostly microbiome, skin microbiome work that I'm doing that's kind of exciting and fun that we're working on. And then I'm also trying to look at how can we use AI, and I will jokingly say that I can spell AI, and that's about as good as I am at the moment, but I'm getting better. But how can we use it for biosafety management? In the US, we have most of the labs that are seen as kind of not transparent, and we tell the public to trust us, but we now need to move to a phase of where we show the community that we're doing the work that we do safely and that we don't have a lot of things to concern ourselves around. And then I do also still do some field work as well. So I wanted to switch over to AI and what AI can and can't do. It's a buzzword. It still is a buzzword, but it really is changing dramatically. And I don't have the time to tell you about the history of this, but about a year and a half ago, I was involved with looking at AI, large language models specifically, and how they were moving and how they could actually solve biological problems. And what it came to be is that this idea of moving from the COVID reality, which is almost a year to a solution, a lot of excess deaths and trillions of dollars. There's different numbers placed on this. And of course, productivity loss as well. So a global pandemic, and how do we avoid that? And I'm really a fan of the CEPI 100-day mission, but I also am a realist. Most people say that I'm not really fun at parties because I tend to say that it's going to be harder and realize all the holes that are going to be there. But I do think this is the way we need to be thinking. And we have to think across both national areas, and we have to think about other types of disciplines that we don't normally think about working in this solution. So this is the problem, and for those of you that are in basic research, that's where I started off and thinking, okay, I'm gonna solve the world's problems, and then I realized this funnel here is 10 to 15 years long. It's about $1 billion US for one product to move it to licensure, and then if you're working with a neglected disease, such as Ebola or Marburg, which is what I started working on, this is very difficult to achieve a solution for. And this is where we need to kind of think about how do we move around this. And the first steps in the US was about the animal rule. The FDA, the Food and Drug Administration, came up with this idea of non-clinical studies for diseases in which there's not a lot of human disease going on and that we can't run those tests. And so how do we get things prepared and how do we have solutions on the shelf when we get to the next stage? So for example, with ZMapp, we had preclinical design of that. We had a preclinical cGMP lot of that. It was ready for preclinical testing. We went around for 3 years asking for funding. Nobody wanted to fund the clinical studies. Nobody wanted to fund the phase 1. So when West Africa outbreak occurred, it was available for a variety of crazy reasons and it was used, but it could have been better prepared. It could have been sitting in a preclinical or clinical setting that would have been safer for the patients to start that. And I always put this slide up not to spend a lot of time on it, but remember there's also animal vaccines. We tend to forget about that, animal therapeutics. So that's a problem too because it's just as regulatory, at least in the US, as much regulatory controls that are placed on that as well. So it's a tough model. So what does this mean we have to do with vaccines and therapeutics? Well, there are a lot of cool omics capabilities that are coming online. And this is just a great review looking at the integrated of AI and machine learning tools. And this is a few years old. And actually it started with this paper and some work during COVID with Dean Ho here at NSU in the US that we started thinking about this concept. And he kind of pulled me out of this realm of should we just go through large quantity testing in the lab, and he's like, well, we should look at the computational space and look at that. And so it was really exciting to be part of that during virtual meetings during COVID when we were locked down. And so I think there's a lot of, after meeting with them and thinking about this, there's a lot of steps in the development that we can start to think about. We need to start moving away from animal usage, for example, but we can actually do computational antigen design in a much different way than we have in the past. Typically, we're still just picking the spike protein of some virus or envelope protein and we're inserting it, not thinking about how it's going to be proteolytically cleaved when it goes into the host and how it will be different in different individuals and present it in different individuals. So we have a lot of more capability of thinking about that now than we did before. And it can be done computationally. It doesn't have to be done empirically, which is really exciting. And then all the way at the end, we have these adverse events reporting in the post-marketing in the US that we have a treasure trove of real human, real life data that can be looked at, and you can go back and find better solutions going backwards. So this is possible today. Is it being done? Not really that often. I just point that we need to move away from animal models. That's one of the common themes. Unfortunately, animal models lie to us. They don't always give us the right answer. So we're looking at alternatives. We're looking at ex vivo systems, organoids, and organs on a chip are some of the new areas that we're looking at. And this can be integrated into AI and machine learning tools. Again, it's kind of the same thing, moving from 2D culture to 3D cultures, organs. Even organoids right now are still kind of 2D, they're not 3D. So there's a lot of capability that still needs to be developed, and the new generation can help us solve some of those problems. And this is a big area that I work with in oncology and metabolics, but also something we need to do in infectious disease is we need to start using our animal data and our human data and real life data that we have. Unfortunately, most of this is locked up and not on data lakes that we can use to actually pull this data together. But you can do a lot of cool things with digital twinning. And this is a capability that AI brings to us now and machine learning tools give us, and we need to start thinking about this a lot more. At least in the clinical trial area, this can really help design the clinical trial stages and actually find responders and non-responders for oncology. And unfortunately, in medicine, we still, infectious disease, we still consider treating everybody the same when we know that that's not the solution. Just like in oncology, there's responders, there's people who have some response, and then there's people that have no response. But yet we still treat everybody the same way, and we give the same drug in infectious disease, mainly because of cost, but we need to really think about that. So how can we move to precision medicine in infectious disease? And we can do that through digital twinning and real data. So I get asked this question a lot about AI. This is a shorter version of this, but will AI solve our problems, the current versions of AI? No. It's a good change in the time that it takes. It actually decreases the time that we need to do some of the work we need, for example, computational antigen design. But there's other things that come into play, such as cost. It really doesn't change the cost of this production system yet, and it doesn't really reduce the risk. So when I'm asked these three questions from a clinical development of a product, it's all about time, cost, and risk. And when I talk to investors, investors always want to know about risk. They don't really care about the cost and time too much, but they know that there's less than a 1 to 5% chance that a product from discovery will make it to licensure. So anything that reduces that, just small percentages, makes a big difference for them. What I will tell you, though, is that agentic AI, which is something that people are just starting to understand, is where things are, and is where I want to focus on the last few minutes of my talk. Agentic AI is not like traditional AI. It uses some of the traditional AI tools and large language models, and then we can use predictive AI, that's using the real digital data and digital twinning. But agentic AI can actually do things that we can't do. And I was on a panel assessing some of these tools in the past, these large language models, and we're asked to say, is it a graduate student, is it a colleague, or is it a high school student? We all agreed towards the end of January last year that they were all working at the expert level. But there was very little novelty into it. It doesn't tell us anything we don't know if we did a large data search or we read the literature. But agentic AI can actually give us novel insights. So agentic AI tools will transform what we're doing, and it actually changes, and this is a review I'm working on at the moment, but it changes the time, it changes the cost, and it changes the risk extremely. So these are where we can get some real cost and time advantages to this as we move forward. So what is agentic AI? Agentic AI is actually using large language models, but actually having it use tools at the same time. So tools can be like AlphaFold, it can be Rosetta, as you'll see in the example I'm going to give you here. So any tool that's out there. So the large language models that currently exist don't understand genetics very well. It doesn't understand the sequences. It doesn't understand amino acids very well. But if you go out and have it look at a tool, and that tool actually can manage that and bring it back as information, it can use that information. So that's what agentic AI is capable of in science right now. And this is where there's a lot of potential. A lot of the US-based large language models are focusing on biology because there's such a huge potential here. So this is the first real paper that I found that I thought was a real game changer, and it was by James Zhou's group and at Stanford. And what he did is basically made some SARS-CoV-2 nanobodies, and not only did the computational side, but had the wet lab work done as well. And this went on to be published here more recently in the last month in Nature. And what was exciting in the Nature paper compared to the early paper in November was that he actually had refined it even more, where the agents did a lot of training. So what he did is he basically had a human user, which was him and a few others, and they developed agentic AI people. So these are all, individuals that are acting inside the large language models. So they had a PI, scientific critic, they had an immunologist, machine learning, and a computational biologist. In the Nature paper, he went further, basically having them become specialists, reading everything that was in PubMed and then taking a test. So it's really impressive how he did that. And then what he did is he had the data sets that he could add into it as well, but they had meetings. And these meetings could be held, you could hold 1,000 meetings, and you could come up with the top ranking ideas. And that's kind of what, in short, what happened. And what it did is it said that they should use an ESM model, an evolutionary structural model. They should then use an AlphaFold and then go to Rosetta to do their finalization and then do some iterations around that. And that's what the computer agents decided was the best choice. And they went and took that information, came up with their sequences, put it into the nanobody actual wet lab work, and then they measured by ELISA to 3 of the isolates from COVID that basically gave them a span of the different variants. And so in that work, what he showed was that they could actually get binding to both the original Wuhan isolate, but also some of the other isolates that have been more divergent over time. And these are same antibodies, these are the same nanobodies that were developed for this one round. They had to do some modifications, and the agents did that modification, and then they went back in the lab and made those. But a really impressive use of agentic AI and a different workflow than the people working on it at the time thought they could do. This has been done in other areas now. There was some de novo design of proteins for snake antivenoms that have been developed in a similar manner. And I forgot to put the last slide up, was a slide on some of the other things that he's done more recently, where he's actually looking at single cell analysis, where he takes the data of other individuals and then sends them the data of how he's actually made these long work paths using agentic AI and can give them new insights to their findings from single cell analysis. So we'll stop there if there's any questions. Again, I'm sorry it's a short talk because it's only like, but it's usually about an hour-long talk on this topic.
I have a general question. This agentic AI can actually understand the quality of a paper in PubMed, or it's just taking everything?
I'm sorry. Yeah. So it does it.
The quality.
Yeah,
because,
the quality.
I
yeah.
mean,
you could actually
face it.
Yeah.
There is a lot of rubbish that is published.
And that is the problem with the large language models. It has biases built into it already and has ideas around certain areas. Yes, you could select papers. I don't know. I have to go his recent paper.
You means who? Me or the AI?
Oh, you could actually come up with a design for that. You could say it's got to be in this tiered journal. It's got to be H factor of X.
Yeah, but also that.
Yes. Yeah. Good question though. Yeah. It's interesting, if they fail their test, if they fail their prelims, they have to go back and retake and do more learning in their specialty. So.
Okay, nice talk. I know you are a supporter for the replacement of the animal models. Yeah. An NIH director, everybody is talking about. Do you think really we are ready to replace the animal models? Maybe one day AI is here. I think it's good for us to play around with AI, the cell-based experiment. If AI design any vaccine without animal models, do you really think we are ready, the whole world, we can get into the human beings? Thank you. That's your opinion.
No, it's a great question, whether or not we're ready. Ready by what time frame? So I think for toxicity studies, we are ready to convert over to some alternative methods for tox. Are we ready for antigen assays and for therapeutics and live models? No. We're looking at more than 20 years probably of getting to that level where we have some type of in silico assessment system, but it's way off in the distance.
Yeah, exactly. Why I'm asking you this question. Maybe we are ready for the AI-based drug discoveries. Knock to your head, you have a drug. Yeah. But that's only for the cell-based, like my suggestion. So my worry is about, when you put it into the human beings, you may need really come back to the animal model again. Yeah. So of course, at the moment, we are using too many animal models. It's too much money, use the animal model. But if you give up animal models, maybe one day we will have so many people sacrifice their life because early test is a human being.
Yeah. I agree. Yeah, okay. Completely agree.